Professor Gopinath, Padma Shri Dr. Gopinath for inviting me to this conference, and Al Jopa for organizing this conference. So the major focus of research in my lab is on estrogen receptor P53 signaling crosstalk in different type of cancers, specifically like luminal and triple negative, breast cancer, high-grade serous ovarian cancer, and non-small cell lung cancer. So in all these projects, the basic idea is to translate basic research to clinical trials and patient care. As you know, breast cancer can be divided mainly into four different subgroups: luminal, A and B, and HER2 enriched and basal-like tumors. So in terms of molecular characterization, these tumors express both the luminal breast cancers express estrogen receptor alpha, whereas the HER2 and basal-like triple negative tumors do not. However, estrogen receptor beta, another member of the steroid superfamily, receptor superfamily, is expressed in all these forms. Another major player is tumor suppressor P53. As you can see here in the luminal A and B, the P53 mutations are relatively low, 12% and 29% respectively. However, if you look at the HER2 enriched cancers and basal-like cancers, there is very high frequency of P53 mutations. In the basal-like, it is almost 80+% of them have P53 mutations. Now, when it comes to cancer, estrogen receptor alpha or ER alpha, it's also called ESR1, is considered as a villain, and P53 is considered as a hero. However, in normal physiology, both these proteins are essential for proper homeostasis. Somehow, in diseases such as cancer, this homeostasis is somehow disrupted. A few years ago, we showed that estrogen receptors have dual roles. In the case of estrogen receptor alpha or ESR1, this is well known. It is in a conventional view of the receptor function. It, in presence of estrogen, it binds the, when ER alpha binds estrogen, it recruits activators and activates a whole bunch of pro-proliferation genes, and the opposite happens when it is anti-estrogen such as tamoxifen. Co-repressors are recruited and pro-proliferation genes are inhibited. What we showed is that not only estrogen receptor alpha can target ER responsive genes, but it can also target tumor suppressor P53 target genes by protein-protein interaction, as you can see here at the bottom. And when it binds to P53, ER alpha, contrary to the conventional pathway, it recruits co-repressors and thereby suppressing tumor suppressor genes, whereas in the presence of anti-estrogen such as tamoxifen, the interaction between estrogen receptor alpha and P53 is disrupted, thereby reactivating tumor suppressor function of wild-type P53. Now, we have shown in a prospective window of opportunity trial that whatever I showed you indeed really happens in patients. I would not have the time to go into that because the main focus of today's talk is the triple negative breast cancer where estrogen receptor alpha is not expressed. Whereas it expresses estrogen receptor beta, and instead of wild type, as I showed you in my second slide, 80% of them contain express mutant P53. So what we showed using a very sensitive in situ as a proximity ligation as a or PLA, estrogen receptor beta and P53 bind together or interact both in the P53 wild type expressing and mutant expressing, which are the majority of triple negative breast cancer cells. And we confirmed that an independent different method called co-immunoprecipitation assay is shown on the right. And this is true in situ in real patient tumors as well. As you can see, on the right, two panels shows immunohistochemistry. On the left, on the right, you can see the proximity ligation assay to show the interaction. And the very last panel, you can see very nice purple dots, which indicates these two proteins are in a complex in these tumors. And further, we went ahead and showed that at the molecular level, these two receptors directly bind each other, and the DNA binding domain of estrogen receptor and the binds to the very C-terminal domain of P53. P73 is another family member of the P53 tumor suppressor family, and P73 can regulate many of the P53 target genes. In P53 deficient cells, P73 can compensate for the loss of P53. However, the problem is that mutant P53 is smart enough to bind and inactivate this tumor suppressor. That's the challenge in triple negative breast cancers and many other breast cancers where mutant P53 is expressed. And so disrupting this mutant P53, P73 interaction has been proposed as a viable therapeutic strategy. And what we have shown in cell culture model system and in animal models based on a whole array of experiments, this is what we know. Like when in the P53 wild type context, estrogen receptor binds P50, wild type P53, inactivates it, therefore it is pro-tumorigenic. However, if P53 is mutant, what happens is that estrogen receptor beta sequesters P53, mutant P53, oncogenic mutant P53 away from the tumor suppressor P73, thereby activating anti-proliferation pathway. So in that context, it is estrogen receptor beta is anti-tumorigenic. So we are providing an explanation of the controversy in the field where estrogen receptor beta has been reported as either, in some reports it is pro-tumorigenic, in other reports it's reported anti-tumorigenic. So what we are showing is that both camps may be correct, but neither camps looked at the status of P53, and that is the determining factor. That's what we have shown in our work. And that was corroborated by bioinformatic analysis of a large breast cancer data bank called MetaBrick, and there it's a mutant P53 breast cancers. We stratified them into high ESR2 expressing and low ESR2 or estrogen receptor beta expressing groups. As you can see on the blue line, the ESR2 high expressing P53 mutant tumors have better prognosis. And then subsequently we asked what's the effect of some of the ligands, the main ligand being tamoxifen, for example. So when you use tamoxifen, what you can see here is that estrogen receptor beta P53 interaction is increased on the right panel here, and whereas reciprocally estrogen receptor, mutant P53, P73 interaction is decreased. You can see the purple dots are lower there. So that's the oncogenic interaction, that's disrupted. And that is reflected in the transcriptional profile in these cells. Like as you can see here, that prototypic P53 target genes, PUMA and P21, both are highly expressed when tamoxifen and estrogen receptor beta are present. You can see the yellow shaded. Similarly, not only estrogen receptor beta, tamoxifen, but also P73 is necessary. When you knock down P73, that effect is decreased. So only when all three are present, this effect happens. Can you forward the slide? It's not moving. I'm clicking, it's keep remaining there. Okay, so then we ask the next question. Can estrogen receptor beta p53 crosstalk can be exploited for new therapeutic strategy against TNBC? And as you know that triple negative breast cancer, current standard of care is that adriamycin, cyclophosphamide, and Taxol or ACT regimen. And doxorubicin is a DNA interacting agent leading to the blockage of DNA replication and transcription. And P73 is responsible for initiating apoptosis in response to doxorubicin in P53 deficient cancer cells. So, however, although this drug is a powerful drug in the clinic, cardiotoxicity is a major clinical problem for this drug. It is dependent on the cumulative dose of doxorubicin. Therefore, the patients cannot be put on this drug on long-term basis. So what we have shown is that when you combine tamoxifen with the doxorubicin, tamoxifen lowers the IC50 of doxorubicin. As you can see, the red curve, the curve is shifted to the left, which means lower IC50. However, when we use the same type of cells or isogenic cells, the only difference being when we knock out P53 using CRISPR technology, then you see that shift is lost. So this means that tamoxifen is targeting the oncogenic function of mutant P53. When mutant P53 is not there in a particular tumor, adding tamoxifen does not have any advantage. So this is very important in terms of stratification of these tumors in selecting what drugs will be effective in the patients or not. So this is also, as I stated previously, that when you have this combination of tamoxifen and doxorubicin, anti-proliferation genes are upregulated. When you knock down P73, SIP73, then that effect is much lower. Similar, so P73 is necessary. There also P53 is necessary. When we knock out P53, all you need to look at is the last lane in each of these two panels, the top. You can see the dark band only appears in the mutant P53 cases, whereas in the P53 knockout case, whether you add tamoxifen or not, the apoptosis occurs. Subsequently, we molecularly characterized this in terms of global gene expression by RNA-seq. I wouldn't have the time to walk through this in detail, but all you need to do is to look at the middle panel, the green panel. When we have the combination of tamoxifen and doxorubicin, 692 genes are upregulated, 785 genes are downregulated. These are unique sets of genes. They are not there, only very small number of genes are affected when you have just a TAM, which is not unexpected because triple-negative breast cancers, tamoxifen is not known to have much effect, but when we combine, there is a huge difference. So that's what is novel here. Okay. So we showed that both in vitro and as you showed in vivo in the mouse models as well. When you treat, the combination has far better effect in terms of reducing the tumor size. And not only that in xenograft, but in patient-derived xenograft, triple-negative breast tumors, you can see the same effect, the purple bar line if you look at, it is much more effective. And whereas if P53 is not mutant, that effect is not there. And now the model is here, like as I showed you, I wouldn't have the time to walk through, but quickly, this is what I showed you happens. And not only that, the tumor extrinsically also this have an effect. More recently we have shown, but I would not have the time to show you that data. But what is important, I just wanted to stop you by sharing two more slides here. In a recent collaboration with clinicians at Harvard and Massachusetts General Hospital, We tested this in a patient. There's a nice story behind it. I'll tell you, I won't have the time to tell this because the patient called me out of the blue and thanked me because she saw our JNCI paper in 2019 and convinced her physician to include tamoxifen in her regimen because she has 15 lesions in her brain and she has nothing to lose. So after tamoxifen, including tamoxifen in the regimen, you can see the bottom panel, the lesions are considerably reduced. And we did the immunohistochemistry to show that the P53 interaction increases after the tamoxifen treatment in these tumors. So conclusion, especially for the triple negative, tamoxifen enhances beta mutant P53 interaction, leading to the reactivation of P73. Tamoxifen lowers the IC50 of doxorubicin. Combination of tamoxifen and doxorubicin enhances cell death dependent on both estrogen receptor beta and P73. Co-expression of beta and mutant P53 should be an important prognostic factor for TNBC. Precision medicine in terms of repurposing of tamoxifen with the chemotherapy regimen to treat a molecularly selected group of TNBC could be feasible. This is especially relevant in developing countries like India because tamoxifen is a well-known drug. It is a low-cost drug, not like many other new therapeutic agents. It is very affordable to most people. So doxorubicin dose much lower than that is what is currently used, can be used if this is successful, lowering the effect of doxorubicin, including cardiotoxicity. Tamoxifen is inexpensive, and the treatment can reach the patients relatively fast because it is repurposing, potential to combine with immunotherapy also. So based on that one patient case, as a scientist, I cannot say that one patient is big, but in clinic they say that it is big. So we are now starting a clinical trial on larger number of patients between Roswell Park, Harvard MGH, and the University of Rochester. And finally, these are the players who really made this all happen. I'm just representing them to give this presentation. Thanks goes to all of them.